Hello and welcome to our poster presentation titled idg.reactome.org, a web-based platform for visualizing dark proteins in the context of reactome pathways. My name is Tim. I'm a member of the Reactome team, and I'm going to show you around. Reactome provides a pathway-based approach for visualizing biology. Pathway diagrams, a key feature of the pathway browser visualized here and here on our poster, allow researchers to visualize complexes, proteins, reactions, and more in the context of biological systems. The idg.reactome.org portal for the Pathway browser provides extended functionality and some new tools to help researchers uncover the function of understudied or dark proteins. By definition, for us at least, a dark protein is defined as a protein with no known drug targets or small molecule associates, and with no or few publications. If you'd like to hear more about the data backing this portal, you can visit a poster presentation given by Naseem Sanadi, another one of the Reactome team members, and her poster is titled Expansion of Reactome Functional Interaction Network to Allow Exposure of Knowledge Space of Understudied Proteins in the Context of Biological Pathways. In this presentation, I'm going to give you some insight into the new features that we've added to the Pathway browser. First, we've added a functional interaction view that allows researchers to visualize any pathway diagram in a set of functional interactions as a network. We've also added the ability to overlay tissue-specific expression data from the Target Central Resource Database and the ability to overlay pairwise interactions from a number of different sources. So I'm going to start with the tissue-specific expression overlays with this form here. Here you can choose one of 19 expression types present in the TCRD database and up to 12 tissues to display at a time on pathway diagrams and the functional interaction view. On the pathway diagram here, you can see the default view without an overlay, and this shows the target development level of each protein and each protein inside of a complex. Here you can see a red T dark protein. This is an example of an understudied protein with no or few publications and no drug targets. Here you can see a tissue-specific expression overlay where the complexes are recolored so that each protein's expression is displayed for any specific tissue. If you'd like to visualize all of the proteins in a complex and all of their expressions across multiple tissues at once, you can right-click on a complex, which will open this Entity Detailed panel here. Here you can see listed all of the proteins and their expression value for every tissue selected. Similarly, you can get to this panel from the functional interaction view by right-clicking on any one of the protein nodes. Next, I want to talk to you about overlaying pairwise interactors. Here we have the form that allows researchers to select sets of pairwise interactors. And we have a few different types, including protein interaction, gene co-expression, gene similarity, and some other relationship types as well. You can also choose the data source for this interaction type, as well as, in some cases, different biosources and tertiary options. For each set of interactors, we allow users to pick a positive and negative line color so that they don't get confused when looking at the pop-up I'll demo in a minute here. You can add up to 6 sets of interactions at once to visualize on the pathway diagram and in our pairwise pop-up. So how do you open this pairwise pop-up here? Right-clicking on these red circles here will open the pairwise pop-up window for the entity selected. In the pairwise pop-up, you'll notice a functional interaction network at the top here. The circular nodes in the functional interaction network represent the diagram source proteins of the entity that you opened. The triangles represent the functional interactions displayed on top of the source nodes here. You'll notice that both the source nodes and the interactors are recolored to match whichever tissue-specific expression you have overlaid at the time. Below this functional interaction network, you also have a table available to you that lists all of the pairwise interactors, their current overlay value, whether or not they're a positive or a negative interaction, and the source that the interaction came from. There are, in a lot of cases, too many interactors to display in the Cytoscape view here, or the functional interaction view, as we call it. And you can add ones that aren't displayed by clicking this view button. If there is an interactor that you don't want to display in this view here, you can right-click on it and remove it. One of the other things that we've added is the ability to overlay drug-target interactions onto the pathway diagrams. Here you can see in this diagram this purple circle in the top left corner. Clicking on that will open the same panel, and you'll be able to see these purple hexagons here representing drugs with edges connecting them to their source interaction node. You also have a table of this data below the interaction network. You'll notice that you can open both of these windows for one complex at the same time. So you can have drugs and functional interactions at the same time in this network view here. You can also open this pairwise pop-up by right-clicking on a node. You'll get this same entity detailed panel, and you can open it with this button here. Similarly, for drug-target interactions, you can display them directly on the functional interaction network through an options menu. If you have any questions about our work on the idg.reactome.org portal, you can visit us at reactome.org or at idg.reactome.org. Thank you for attending our poster presentation today, and we hope that you enjoy the conference.